All right. So in this lecture, I want to specifically address kind of a point of confusion, and that is answer to the question, what is energy? Okay, so typically when we talk about energy, we talk about cellular energy, and we're talking about ATP, or it's misconstrued as just ATP. But actually in a cell and cellular processes, there's a lot of different forms that energy can take. We've already talked about energy being stored in the form of chemical bonds as a type of potential energy. There's a couple other common types of cellular potential energy. And I just kind of want to address this in this part of the lecture. So whenever we're talking about energy transformations, what are we talking about when we're talking about that energy? All right, so returning to our metabolism lecture. So something that's really important to note here is that whenever we're talking about energy, for the most part, we're going to be talking about energy in the form of electrons. So whenever we're talking about moving energy in and out of chemical bonds, really what we're kind of talking about is electrons. So when electrons are kind of utilized in chemical bonds, these can be stored in chemical bonds, and in that case, if they're being stored in chemical bonds, anytime any energy is stored in a chemical bond, that's what we are talking about when we're talking about the potential energy that's stored in a chemical bond. It has to do with the fact that we have electrons involved in the formation of that bond. So whenever we're talking about chemical bonds being a form of potential energy, what we're talking about is that we have electrons which are stored within that bond or are used in the formation of that chemical bond. Why is this stored and inaccessible energy? If you've probably studied in physics or chemistry, electrons in general have a lot of kinetic energy. So if we kind of like use them in the formation of a chemical bond, they're not as kind of moving around as much. They're stored and kind of held in place. However, if we want to use the energy in the form of electrons, what we can do is we can break a bond and release electrons. And if those electrons are released, they're free to kind of move around. They're free to use in the formation of other chemical bonds, and therefore freely released electrons would be a form of kinetic energy, usable energy. So this is one of the primary kind of forms of energy that we're going to be talking about when we talk about metabolism. We're going to be talking about how we're moving electrons around. So again, electrons are primarily stored in chemical bonds or utilized in the formation of chemical bonds. So we'd say that they're stored in chemical bonds. And if we want to release those electrons for use in other chemical reactions, we would have to break that bond and release those electrons. One bond that is extremely important in both its formation and its availability is a bond in the molecule that I'm sure you are all familiar with, called adenosine triphosphate, or ATP. It's called adenosine triphosphate, or ATP, because it is composed of a molecule called adenosine, and then it is composed of another molecule, ribose, sugar. And then attached to that ribose, there are 3 phosphate groups. That's why it's called adenosine triphosphate, is because there are 3 phosphates attached to that sugar. And because of the way that these bonds are formed, kind of the stability of these bonds, what happens is the bond between the 2nd and the 3rd phosphate on adenosine triphosphate is able to form what's called a high energy bond. So this has a lot to do with what we're not going to talk about in this class. So you can ask your biochemistry professor. when you get there. But because of the electrochemical properties of this bond, this is a very energetic bond. But what that means is that when ATP kind of gets to the area of a chemical reaction, it is very willing and able to break and allow the electrons stored in that bond to be utilized in other chemical reactions. So ATP is, you know, we think of it as sort of like the cell's preferred source of electrons or source of energy, because it has that really high energy bond between the second and the third phosphate in that molecule. So ATP actually serves as sort of like a rechargeable battery in the cell. So we can, basically the goal of a lot of your catabolic reactions is to release energy from other bonds and use that energy to create ATP. So ATP can be created if you take adenosine and a ribose and two phosphates, what would be called adenosine diphosphate, and then add a bond between adenosine diphosphate and another phosphate to make adenosine triphosphate, which is what you're looking at here. ADP, adenosine diphosphate, plus a phosphate would generate ATP. That is a chemical reaction that is mediated by an enzyme, and that enzyme is called ATP synthase. So again, like we talked about, that is a very high energy bond. So that means we have to invest a lot of energy into its formation. So you can see that we get that energy primarily, we get it from a couple of places, but we can get a lot of that energy via the process of cellular respiration. Well, essentially what we do with cellular respiration is, as we will talk about later, we use an organic molecule, glucose, for example, which has bonds between 6 carbons and some oxygen and some hydrogen as well, lots of bonds in that glucose. And what we do is we break those bonds, release electrons from those bonds, and harness all of the energy that is stored in those bonds via those electrons, and kind of use all of that energy to form as much ATP as possible, to create as many of these high energy bonds as possible. So that energy has to come from somewhere, right? We can't create it, we can transform it. So what we're doing is taking potential energy stored in organic compounds, releasing it in the form of electrons, and then storing it in sort of a higher energy bond, a more accessible bond. And then we can use that energy stored in that bond to do other chemical reactions. So that's kind of, but when we use it, we break that bond. We are left with ADP, so we can use cellular energy to kind of like recharge that battery into ATP. So the nice thing about ATP is it is a really, really good source of energy for cellular work, for fueling its metabolism. But once we use it, it's rechargeable, right? Cellular respiration serves to recharge the battery, that is ATP. So since ATP is so extremely important to a cell, there's a lot of ways you can make it. So we can divide the ways that we can make ATP into 3 primary mechanisms. These are called 1, substrate level phosphorylation, 2, oxidative phosphorylation, and 3, photophosphorylation. So what I'm going to do with the rest of this lecture is just serve to kind of talk about how these 3 processes take place and differentiate them. The biggest difference here, okay, right? Every single one of these, all 3 of these, substrate level, oxidative, and photophosphorylation, all of these are their goal is to create a bond between ADP and a phosphate to create ATP. That's the goal of all 3 of these. The difference between them is where we are getting the energy to create that bond, okay? It's the big difference. And you can see that in the bottom right here. The big difference is the source of energy used to create that bond. So let's go through all three of these. So the first of these is called substrate level phosphorylation, or SLP. So in order to remember this one, what you need to realize or remember is what the term substrate means. So recall that a substrate is whatever is being acted upon by an enzyme, or you can think of a substrate as a reactant in a reaction. It's what's going into a reaction. So what this should tell you is that the substrate itself is releasing the energy required to generate ATP. Okay, what the heck does that mean? So remember that as far as the energy we're talking about in this class, one of the primary forms of energy we're talking about is electrons. And remember that electrons are stored in chemical bonds. So a substrate level phosphorylation, what happens is that, well, there's two alternatives. There's two things that can happen. You can have a substrate, right? And remember that you may have, if you're going to be breaking a bond, that substrate has a bond. And what happens is if you break that bond, that will release energy. And that could be enough energy to fuel the creation of a bond between a phosphate and ADP, right? So there is energy in the form of electrons released directly from a substrate, all right? Substrate level phosphorylation. Whenever you hear the term phosphorylation, adding a phosphate to a molecule, such as adding a phosphate to ADP to create ATP, that is called phosphorylation. So whenever we are converting ADP to ATP, we are phosphorylating ADP, all right? So in the case of substrate level phosphorylation, the energy that we need to create ATP is coming directly from our substrate. So as I mentioned, this energy could be that we're breaking a bond, releasing electrons, and those electrons are what's going into the formation of that bond, okay? Alternatively, substrate level phosphorylation could also be if we have a substrate that has a phosphate group associated with it, and then when we break a bond, that phosphate group is released and attached to an ADP. So here, it doesn't really matter what we're talking about, but what we're getting with substrate level phosphorylation is either, or and, both the energy to create this bond, or the energy and an actual phosphate to create that bond. Okay, so the big thing here, substrate level phosphorylation, this is creating ATP, phosphorylating ADP, and you're getting the energy to phosphorylate ADP to create that bond directly from a substrate in some sort of chemical reaction, okay? Or you're getting the phosphate from that chemical reaction. All right. So let's move to our second type of phosphorylation, and this is called oxidative phosphorylation. This is a little more complicated, involves a few more steps, but it is really simple once you understand kind of the terms. So just like with substrate level phosphorylation, this tells us exactly where our source of energy comes from. And in this case, our energy in the form of electrons to build that bond, to phosphorylate ADP and create ATP, our energy comes from the process of oxidation. Okay, so this is where we need to get into a little bit of chemistry. So just to remind you, or to reiterate, or to give you a preview, regardless, depending on at what stage you are with chemistry. Either you've had it, you're in it, or you will have it, right? There are reactions. These are paired reactions, and these are called redox reactions. So redox stands for reduction reactions paired with oxidation reactions, or oxidation reduction reactions. However you remember it, Redox, read, is referring to reduction, and ox is referring to oxidation. These terms are referring to what is happening to electrons associated with a molecule. In oxidation, when a molecule is oxidized, electrons are released from a bond. Okay, so if we look at this picture right here, we have a molecule, a bond is broken, electrons are released. This molecule, because electrons are released, this molecule is oxidized. Okay. In a cell, you don't really want electrons just being like hanging out. Electrons can do a lot of damage. And so the reason oxidation reduction reactions are always paired, the reason these are redox reactions and not just oxidation reactions, or if you've heard of like oxidative damage, is because just free electrons are not good. They can go around and just like terrorize other chemical bonds. So oxidation reactions, when electrons are released from a chemical bonds, are almost always paired with reduction reactions. And reduction reactions are when another molecule grabs electrons, receives electrons. So with redox reactions, you have a molecule where electrons are released, it's oxidized, electrons are released, and then you'll have another molecule that is reduced, meaning it gains those electrons. A molecule that gains electrons is said to be reduced because electrons have a negative charge. So whatever molecule receives those electrons, its charge is reduced. So that's why we say it's a reduction reaction. So again, a redox reaction is paired reaction, two reactions. Oxidation is when one molecule is oxidized, meaning it's releasing electrons, and then reduction is when another molecule is receiving those electrons. It's reduced. So with oxidative phosphorylation, obviously we're moving electrons around. We are releasing electrons from a bond, and then those electrons are being grabbed up by something else. Something is being reduced. But in the terms of how we generate enough energy to create ATP, in the form of oxidative phosphorylation, those electrons are actually carried to a membrane, and then they will eventually help in the formation of another form of cellular energy. And the cellular energy is what's called a proton motive force. So we're going to talk about on the coming slides. So it is important to remember that notice that substrate level phosphorylation, the energy required to generate ATP occurs right there. Okay, a bond is broken, energy is released, you get ATP. Easy peasy, okay? With oxidative phosphorylation, and as we will talk about with photophosphorylation, there's actually two steps. So with oxidative phosphorylation, what happens is you have a redox reaction. You have a molecule that's oxidized, you release electrons. You have a molecule that's reduced, it gains those electrons. But then the second part is those electrons are actually carried to a different location in the cell, a membrane, and they are used to generate another really, really potent form of cellular energy, and that is what's called a proton motive force. Okay, so first off, just to review what we said about redox reactions. Redox reactions are paired oxidation reduction reactions. That's where we get the redox. You can remember this via the anagrams. I always was taught oil rig. Oil oxidation is loss of electrons. Rig reduction is gain of electrons. One of my colleagues taught me this one, which I had never heard, but you might like it. Leo the lion says Gur. Leo, loss of electrons is oxidation. Gain of electrons is reduction. However you want to remember it. Just know that when a molecule is oxidized, it's releasing electrons. That means that a bond is being broken. And anytime a molecule is reduced, it's gaining electrons. okay? Typically that means a bond is formed. All right, so this is kind of looking at this, right? If you start with a reactants, you have your A as a reactant carrying an electron, plus B. What can happen is a reaction can take place, and A releases the electron, is oxidized, and B accepts that electron, and therefore it is reduced. So that's oxidation. A is oxidized because it lost an electron. B is reduced because it gained an electron. Right. So this is kind of looking at redox reactions. So a little bit different here than what we were looking at with substrate level phosphorylation. So remember, with substrate level phosphorylation, we have a bond. We break that bond. Electrons are released. ATP is generated, right? ATP is generated because ADP is reduced. Then we get ATP. That's substrate level phosphorylation that happened at the level of the reaction. Redox reactions are, like I said, are a little bit different because we have a couple of different steps. So here, what you can see is you can have a molecule. That molecule's bond is broken. When you break that bond, electrons are released. So this molecule is oxidized. But what happens is you would have another molecule, in this case like X, accept that electron. So it would be reduced. So here's something else that is really important to note with cellular reactions. Typically, if something accepts an electron and its charge is reduced, typically the body wants to kind of like neutralize everything. So if something gains an electron and is reduced, it's gaining a negative charge. You're going to have something else to like balance out that negative charge. Hydrogen ions, which are basically just protons, are really good at balancing negative charges. So you can typically see if something has been reduced in an oxidative reaction, if you see that it gains hydrogen, actually. So I can tell in this reaction that we start with this reactant, right? And it has a bond. We break that bond, release electrons, so it's been oxidized. And we can see that X is reduced. It gains an electron, and I can tell that because it also gained a hydrogen. It gained a positive charge to balance out the negative charge it grabbed when it grabbed that electron. So if you've heard of cellular respiration before, you've probably heard of these electron carriers. We're going to talk about them a little bit later, like NAD and FAD. Those are kind of the stand-ins for this X here. So a good example of this process of oxidative phosphorylation is whenever you go through cellular respiration, glucose is actually oxidized. So you have the bonds in glucose broken, you're releasing electrons from those bonds. Those electrons are bound by NAD. These are called electron carriers. So what happens is NAD grabs an electron, but in order to balance it out, it also grabs a hydrogen. So you can see, again, that NAD has been reduced because when it grabbed that electron, it also grabbed a hydrogen. So if you see a molecule gain a hydrogen, it typically means it also grabbed an electron. Okay, so we're going to talk about these electron carriers a little bit later. If you're going to have an oxidation reaction, you're also going to have something that's reduced. Electron carriers in the form of these energy reactions are going to be the molecule that's reduced. Okay, so this is again step one of our oxidative phosphorylation reactions. So we have a chemical, we break a bond in that chemical, we release electrons. We have another chemical, electron carriers that are reduced. Okay, so now they're carrying that electron. Where are they carrying it? Where are they going to take it? And the answer, I'll go back to that other slide. The answer is that they carry it in the form of cellular respiration, and honestly, like most phosphorylation reactions, is they utilize what's called an electron transport chain. So there's a lot of terms for this. Another two terms for this is a proton motive force or chemiosmosis. So all of this is referring to the same thing, by the way, which is, as you can see here, remember I kept saying that membranes are the most important part of a cell. Here's another example of that. You have to have a membrane to generate a barrier. And what a proton motive force is you have energy in the form of electrons, and you also use those hydrogens that were gained. So remember, we look up here, we have electrons carried, but we also grab hydrogens to balance it out. And we get to a membrane, and what happens is both of those are released, both electrons and hydrogens. But what happens is those electrons start pumping those hydrogens to one side of a membrane. Okay, so why is this energy? So we've been talking about energy in the form of electrons. This is actually establishing a whole nother type of energy that exists in a cell, and that is the energy of a concentration gradient. So what happens when you pump one molecule to one side of a membrane is it is a very similar situation to like dammed water. So think about the Hoover Dam, which is on the border of Arizona and Nevada. And think about the fact that the Hoover Dam holds back a tremendous amount of water. So in this case, the membrane is very similar to the concrete barrier that forms a dam. Instead of pushing water back, what's happening is we're pushing a ton of hydrogens to one side. Okay, so I don't really know the like mechanics of a dam, but let's say that like you could open a dam. I don't know, like release a dam, whatever you do to a dam. But what happens when you open a dam is all of that water, which was stored behind that concrete barrier, comes rushing through. That is a tremendous amount of kinetic energy. So remember, potential energy is stored energy, and kinetic energy is usable energy. So what we're able to do here is we have this membrane, which serves sort of like a concrete barrier of a dam. We can use energy in the form of electrons to pump hydrogens to one side of this membrane. That's like using whatever to pump water to one side of the dam. This membrane, or the concrete barrier of the dam, is holding all of these hydrogens or water back. And what we can do in a cell is we can hold all of that energy, all of that potential energy, but anytime we want to use it, we can't. We just like poke a little hole in that membrane. Hydrogens like fly through, but that's flying through, that's kinetic energy, that's usable energy. So electrons are usable energy. These hydrogens that are stored behind this membrane barrier, this concentration gradient of whatever, is also a lot of potential energy. And we can use this by converting it into kinetic energy. So we'll talk more about that later. But again, I want to revisit that this is an example of an electron transport chain. It's called an electron transport chain because it's the electrons that are transporting hydrogens to one side of this membrane. It's also called a proton motive force because hydrogen ions are basically just a proton. So if you're shoving a bunch of hydrogen ions on one side of a membrane, you're generating a ton of potential energy that's in the form of protons. So this is sometimes called a proton motive force because of that. It's a potential energy force in the form of protons. And sometimes this is called chemiosmosis because, why not? No, I'm just kidding. Because if you remember, osmosis is the movement of water across a membrane. So we could also be referring to a concentration gradient. You can get a similar force of energy in the form of like water, which would be chemiosmosis. So all of these are the same thing though. What you're doing is you're generating a really, really, really big concentration gradient, a tremendous amount of potential energy stored behind a membrane. And then whenever you release a molecule, that's converting it into kinetic energy. Again, this is very similar to like a Hoover Dam situation, and then like releasing the water. All right. So with oxidative phosphorylation, what you're trying to do is break bonds, release electrons, so oxidize a molecule, releasing electrons from that bond, having electron carriers be reduced, and when they're reduced, they grab that electron, they also grab a hydrogen, and then carry that hydrogen to a membrane where they pump all those hydrogens to one side of the membrane and generate a ton of potential energy. So the cool thing about this is that in terms of ATP, every one of those hydrogens that's pumped to a side of a membrane equates to 1 ATP. So when it comes to how electrons interact with electron transport chains, the really crazy thing is that, again, every one hydrogen can fuel the production of 1 ATP. So if you look at this, whatever reaction resulted in this proton motive force or this electron transport chain, electrons ended up pumping 1, 2, 3, 4, 5, 6, 7, 8 hydrogens to this side of the membrane. Every one of those hydrogens that comes through this ATP synthase, every one of those hydrogens that comes through is going to be able to fuel the conversion of an ADP to an ATP. So what we're going to do when we go through cellular respiration is we will actually track the amount of hydrogens that can be pumped to one side of a membrane. And if we can track the amount of hydrogens that are pumped to one side of a membrane, this can tell us really easily how many ATP are generated through that process. So just remember, moving forward when we get there, that if we're able to pump 1 hydrogen across a membrane, that will give us enough energy to create 1 ATP. 1 hydrogen, 1 ATP. Okay, so what we're doing with oxidative phosphorylation is, again, this is a 2-part phosphorylation event. First, we have an oxidation reaction that releases electrons from a chemical. Those electrons are bound up by an electron carrier, which also grabs a hydrogen. And then those hydrogens and electrons are moved to a membrane. An electron transport chain is generated. Hydrogen is pumped across a membrane to generate a proton motive force. And that gives us a ton of potential energy, which can be used to generate ATP. So in this case, the source of energy is oxidative reactions in a proton motive force. So again, it's 2-part. But the initial source of that energy is oxidation reactions. Okay. So be able to compare this to substrate level phosphorylation. Remember, substrate level phosphorylation, either the energy or the phosphate is released directly from the site of the reaction. So that ATP would be generated right here. But with oxidative phosphorylation, you have to break that bond, release electrons, reduce another molecule, carry that electron, carry the hydrogen to an electron transport chain, and then generate ATP there. So the important thing, why would you go through all these steps? You can create way, 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 way more ATP through oxidative phosphorylation. So even though it seems a little bit more complex, it's way more efficient. So we've talked about 2 ways to make ATP. Let's talk about the third. Photophosphorylation. So remember, the thing that distinguishes all 3 of these is where the initial source of energy comes from that allows us to generate ATP. And as you can probably infer from the name, photophosphorylation, the energy that fuels the production of ATP comes from this side. All right. So we'll talk about this later when we talk about photosynthesis as well. But basically what happens is that you have electrons released from something, but they're released and energized via light from the sun. Those electrons do end up going through an electron transport chain. So we also have an electron transport chain, a proton motive force with photophosphorylation, similar to oxidative phosphorylation. It's just that the initial source of energy doesn't come from an oxidation reaction. It comes from light from the sun. All right. So again, you have three ways of generating energy. You have one, substrate level phosphorylation. Substrate level phosphorylation is when you have a molecule and a bond is broken. So that's why I say here it's an exergonic reaction, because you have energy released right there. That energy is harnessed right there to make ATP. That's substrate level phosphorylation. Two, you can have oxidative phosphorylation, where you have a bond broken, electrons are released, those electrons reduce another molecule, an electron carrier. Those electrons are carried to an electron transport chain, which generates a proton motive force, which creates a tremendous amount of potential energy, which is used to generate ATP. The initial source of energy is from an oxidative reaction. And then finally, you can have photophosphorylation, which is very similar to oxidative phosphorylation. It's just instead of an oxidative reaction kind of being mediated by an enzyme, an oxidative reaction is mediated by the sun. Like photons are breaking apart something. Photons, light, are breaking a bond, releasing electrons. That results in an electron transport chain and a proton motive force, which will be used to generate ATP. So substrate level phosphorylation occurs right when a bond is broken. You get ATP. Both oxidative phosphorylation and photophosphorylation involve a bond being broken, electrons being released, those electrons being utilized to generate an electron transport chain, which will generate a proton motive force, which will be used to generate ATP. You need to understand the difference between all three of these, and we will be going into cellular respiration photosynthesis in the next mini lecture, and we will be able to have direct examples of all three of these. All three of these take place in certain metabolic reactions. 1 and 2 both occur in cellular respiration. 3 occurs in photosynthesis. All right.